Cora MacAlister from Molecular, Cellular, and Developmental Biology. And she's going to talk to us today about a conserved protein modification with multiple functions in different plants.
Good morning. I just wanted to thank YQ for organizing the symposium. Not an easy task, I imagine somewhat like herding cats, but I'm very glad everyone could come today. As Jonathan said, I'm in the MCDB department, and today I'd like to tell you a little bit about some of the work that my lab has been doing. So we're very interested in sort of evolution in a very broad sense. So the evolutionary biologists among you may be cringing at this very simplified phylogeny, but essentially we're interested in characters that are shared among all of plants that are not found in animals or fungi. So green algae, the sister group to the land plants, diverged a very long time ago from the lineage that led to the extant land plants, including the flowering plants, which are united by the production of flowers, appropriately enough, the production of a protective tissue surrounding the ovule. Gymnosperms share a trait of pollen production with the flowering plants. If you go a little bit farther back evolutionarily, you'll see the evolution of vascular tissue that allowed plants to get larger and form roots that penetrate the soil and stems that reach above the surface of the ground. And then these other relatively smaller groups of organisms, including the liverworts, mosses, and lycophytes. So these all have different traits that they share within members of their subgroup, but they also share another very large suite of traits that unites them and reflects their evolutionary inheritance. And one trait that I will talk about today is something you probably haven't heard of. This is a specific type of post-translational protein modification we call Hyp-ara. And this is one type of post-translational modification. Proteins are subject to a very large suite of potential modifications. These include things like methylation, phosphorylation, the addition of phosphate groups, cysteine bridges, forming covalent links between different residues within the protein, hydroxylation, the addition of hydroxyl groups, ubiquitination, and a whole bunch of other elations, including glycosylation, the addition of sugars to protein backbones. And this Hyp-ara modification that all plants and green algae share is a type of glycosylation. So essentially you start with a protein, which has a proline, one or more, in its protein sequence. This gets hydroxylated. So the addition of this hydroxyl group then serves as a substrate for the addition of sugars. So this O will become linkage. And in the case of Hyp-ara, or Hydroxyproline O-arabinosylation, you'll add a short chain of arabinose sugars onto this hydroxyl group on this proline. So these modified prolines tend to occur in clusters in the proteins that are modified in this way. And those proteins also tend to be highly repetitive. So you'll have these clusters of prolines that get arabinosylated in these repeats regularly. And so by the time all is said and done, if you take one of these proteins that are modified in this way, you end up with a glycoprotein that's more arabinose by weight than actual protein. So the plants and algae that do this are committing quite a bit of sugar and quite a bit of their resources to modifying these proteins very heavily, in fact. So why do they go through the trouble of doing all of this modification? And that's the question, one of the questions we're really interested in my lab. So fortunately, we can use a genetic approach to ask that question. So when you start with this proline, this hydroxylated proline, you add these sugars one by one, and that addition is catalyzed by a specific family of enzymes. So the enzymes that add this first arabinose are conveniently named Hydroxyproline O-arabinosyltransferases. That is a mouthful, so we just call them HPATs for convenience. So once that HPAT has added that first arabinose sugar, another unrelated group of enzymes can add the second, and then another group of enzymes can add the third, and then so on. Okay? So these are non-processive enzymes. The HPATs add the first sugar, and then you have different enzymes that add the later sugars. Okay. So if we take this group of enzymes and knock them out genetically by mutation. You will hydroxylate those prolines. That's done by a different enzyme, but you're not able to arabinosylate them. So you don't add this first sugar, and you don't form the substrate that will be acted on by these later acting enzymes, okay? So in the HPAT mutants, we totally disrupt this process, and you don't have any sugar addition, any arabinose addition onto these prolines. Fortunately, the HPAT enzyme family is actually quite small, and it's very deeply conserved in plants. So Arabidopsis, the model flowering plant, I'll show you some data for Arabidopsis, has 3 members of this family, and that's sort of the typical number across the plants that have been sequenced so far. Physcomitrella patens, which I'll also tell you about, is a moss species that has only 2 of these guys. Okay? So the interesting thing about these is that they're very, very strongly conserved. So if you compare the sequence of these from Arabidopsis to Physcomitrella patens, you have a protein across the entire enzyme sequence that's about 70% identical. So there's very little changes in sequence between the flowering plant and the moss versions of this enzyme. And that's quite remarkable considering how divergent these 2 are. So these 2 are separated by about 450 million years, depending on who you ask. So even though they've had a very long time to evolve independently, there seems to be very strong selective pressure to maintain that protein sequence. So these guys are doing the same thing, and that thing seems to be important for plants. But why? Why do plants go through the trouble of modifying proteins in this way? So to answer that question, we're looking at a couple of different species, including the model moss Physcomitrella patens. So Physco development is fairly simple. If you grow them on nutrient agar media, you'll form this little network. We call it a colony, even though it's really not a colony. And so I do my best in talks to not say colony. But you form this small plant that is composed primarily of this network of tip-growing filament cells. So you'll have cells that are elongating at a single point. When they get to a certain length, they'll divide. The tip cell will continue to elongate, and they'll initiate branch points to increase the number of filaments that's being produced. Eventually, they will form these 3-dimensional leafy gametophores. And if you're walking around outside and you see moss, this is the stage of moss you are likely to encounter. Okay. So what happens if you take the HPAT enzymes in this family and you knock them out genetically? So we have 2 HPATs in moss. If you knock one of them, HPAT A, or both of them, A and B together, out, if you're used to looking at moss, this is actually a very strong phenotype. So here's the wild type, here is the mutant. If you compare the size of that network of tip-growing filament cells, you'll see there's a significant increase in the mutants relative to the wild type. And so you can measure that over here. After 3 weeks, you see this really strong increase in network size, which is apparent even after 2 weeks of growth of these guys. And if you look at a little bit earlier stage, when the colonies are still expanding, when the networks are still expanding, so here's a wild type, you can see it's relatively dense in terms of the filaments that are being produced. And then here's the mutant, you can see these longer filaments that are sort of escaping the central network. Okay, so moss plants without HPAT activity are somewhat paradoxically bigger than their wild type counterparts. Why are they bigger? So if you look more carefully at the filaments that are produced, you'll notice that the individual cells tend to be longer in the mutants than in the wild type. So here we're just measuring individual cells along a filament relative to the initial cell that was formed when that branching event initiated. And you can see that the wild type maintain a pretty steady size. The mutants start out a little bit longer than the wild type, and over time they continue to get longer. Yeah? So the individual cells are longer in these mutants. The filaments are also elongating more quickly. So if you compare the individual filaments' length over time, these are pictures that are separated by 24 hours. This is wild type, this is the mutant. If you grow them under particular lighting conditions, you can either favor this fast-growing type of filament or this relatively slow-growing type of filament. But regardless of which type of filament is elongating, the mutants will tend to elongate more quickly than the wild-type filaments. There's a lot of variation between filaments, but on the whole, in the population level, you have this increase in elongation rate in the mutants. Okay, so the mutants elongate faster, they get bigger. In the case of moss. What happens in Arabidopsis? So Arabidopsis is developmentally more complicated, but if we take the analogy of what we've learned in moss, if we get bigger plants, if we knock out the H-pats, we should go from Arabidopsis to sequoia if we knock out the H-pats. That was a joke. So what happens if you knock out the H-pats in Arabidopsis? So if you compare wild type over here and the H-pat triple mutant, there's 3 of these in Arabidopsis, there's not much difference in terms of vegetative phenotypes. They both form normal rosettes, they both form inflorescences and flowers that are normal morphology. If you look a bit more carefully at them, you will notice that the fruit tend to be smaller in the mutants than the wild type. So here's a nice wild type Arabidopsis salique. It's filled with seeds, it's relatively long. And then the mutants, either the triple or the H-pat1,3 double mutant, have this reduction in fruit size. Why are the fruits smaller? The fruit are smaller because they have a fertility defect and they contain fewer seeds. So if you clear the salique, you can see all of the seeds developing inside. Wild-type Arabidopsis will produce about 50 seeds per salique on average, and it's the growth of the seeds that triggers the growth of the fruit. So if you have the H-pat mutants, where you have fewer seeds being fertilized, so fewer seeds developing, you don't get the same signals to elongate the fruit in the same way. So you get about a 10–20% of the seed yield from the H-pat mutants that you do from the wild type. If you look at the RRAs, these are the enzymes that add the next arabinose, so the H-pats are first, and then the RRAs work, you get a very similar effect. So this seems to be specific for that modification and the production of fertility. So what's going on that's reducing the fertility in these guys? Well, there's a couple possibilities. It could be the pollen is defective. It could be the ovules are defective. If you pollinate the mutants just with wild-type pollen, that rescues the phenotype, which suggests it's a pollen defect. And you can look in vitro at the pollen and see that there is indeed a defect. So if you grow these pollen tubes in vitro, the H-pat mutants will tend to be much shorter than the wild-type pollen tubes. They also have some additional defects. The tubes tend to be wider in the mutants, and you also get this occasional pollen tube branching phenotype, which you never see in wild-type. So there's something sort of structurally weird going on with these guys. So I am happy to speculate on why we have this difference between moss and Arabidopsis. It's likely related to the differences in the proteins themselves that are modified, but in the interest of time, I'll just thank my lab members, including graduate students Roop, Stephen, and Cecilia, my lab manager Ali, a cast of undergrads, and our collaborators. And with that, I'm happy to take any questions.
Questions for Cora?
I don't know whether it's feasible or not, but have you looked at whether the moss growth also branch a little bit more, just like the pollen tube of Arabidopsis?
So we don't see any difference in the rate of branching between those guys. So pollen tubes normally don't branch. And the way some moss do is that so they'll have a filament and there'll be a cell division event that will initiate a new tip cell. That's not what we see in a pollen tube. You see it's more you have a tip cell and then for some reason you form another tip within the same cell. So it seems to be a different defect in that way. But that's a good question.
Okay, I just wonder, what are the client proteins of H-pat?
There's a couple that we know are modified. This includes the extensins, which are cell wall structural proteins. There's also some small signaling peptides that are known to be modified. There's another group of enzymes that are inferred to be modified based on sequence similarity to the extensins. So there are knowns and there are suspected, and then there's a whole universe of potentially things that we that may be modified, but no one's actually tested them for modification. So that's another area of focus in our lab, is actually showing which proteins are modified in this way, not just the superabundant ones that we already know about.
So just one more very short question. So how about the homologs in animals?
There's not, so this modification is not found in animals.
Okay.
Proline hydroxylation is present in animals. For example, it's a very abundant modification on collagen, but that doesn't serve as a substrate for glycosylation. So the H-paths are a plant-specific family, and the modification is plant-specific as well.
Thank you.
All right. Yep. Thank you, Cora.